Hi, everyone. We'll slowly make a start here. My name is Jeroen. I'm the co-founder and CEO at Semarion. And first of all, thank you all for attending our webinar. There might still be a couple of people who will be joining us, but we'll slowly start. And we're quite excited to share with you all what we're doing at Semarion to accelerate cell-based assays, specifically with our SemaCyte Microcarriers. So today we'll run you through how these materials work and how we think they can transform cell assay workflows, specifically to make them faster and to produce better data. But before we delve into the science and technology of all of this, a couple of housekeeping points with regards to this webinar. So the webinar is being recorded. We will be using the recorded version to share afterwards online as well. But everybody's microphones and video feeds have been automatically turned off, so you cannot accidentally put on your microphone or your camera, so don't worry about that. We don't have a chat active today, but you can pop any questions you might have in the Q&A section, which you should normally see at the top of the Teams app. So there's a Q&A button, and then you can pop in a discussion point or a question, and then we'll tackle all those questions at the end of the presentation. So feel free to pop them in there, ask your questions during the presentation, and then we'll tackle all of them at the end. It might be that if you are using the web browser, you are not able to put any questions in the Q&A. It really depends on what kind of software you're using. If you do have any issues with that towards the end, feel free to do the raise hand gesture, and we could potentially activate your microphone if there's a question that you would still like to ask towards the end. These are the topics that we'll be covering today. So first of all, we will explain what SemaCyte Microcarriers are, how they function. Then we'll go into how you can use them in practice, the protocols, workflows, etc. And then we'll highlight a couple of use cases, specifically assays where we think we've been able to make a benefit in terms of the throughput of the assay or the data quality, for example, with our SemaCyte Microcarriers. Then finally, we'll discuss very briefly our early adopter program, where we're providing exclusive access to these materials. So just to explain how you can get access to SemaCyte Microcarriers. And then we'll spend a couple of minutes on the Q&A. And as mentioned before, if you do have any questions, feel free to add them via the Q&A function of Teams, and then we'll tackle all these questions towards the end. These are today's speakers. So I myself will be covering the first section on what SemaCytes are and how they function. Then our cell biologist, Blaise Louis, will talk about how to use them in practice, what the protocols are, what the workflows are, etc. And then Dan Weeks, our cell biology lead, will talk about a couple of use cases, how we've been able to use them in plate reader experiments for CellTiter-Glo, NanoBRET, cell painting, various different types of assays that we've validated. So those are the topics that Dan will cover towards the end. And again, for anybody who's just arrived, if you have any questions, feel free to pop them in the Q&A section, and we will tackle all of those at the end. All right, so let's kick off. As I mentioned, I'll be talking about the SemaCyte Microcarriers and how they function. Specifically, we've developed two platforms: the SemaCyte platform, which allows you to move adherent cells, and a SemaPlex platform, which allows you to barcode adherent cells. So what are SemaCyte Microcarriers? They essentially function as ultra-miniaturized mobile wells, which carry small colonies of adherent cells. These materials can be manipulated with liquid handling tools and are controlled magnetically. They are compatible with various assaying formats, plate readers, and microcarriers. So you don't have to use any additional equipment to use these materials. They're essentially a plug-and-play material that you can add to existing workflows, specifically to enhance your existing workflows, and it also enables you to develop novel assaying methodologies. Cells can be seeded onto these microcarriers with standard culturing procedures and protocols. This is something that Blaise will explain later. And once the cells are loaded onto the microcarriers, they can be dispensed into microplates to enhance specific assays. So the SemaCyte platform essentially enables you to move and free cells while retaining their adherent morphology. On the left here, you can see a graphic of what these materials look like. They have a flat cell growth area, so that cells can attach and grow as they would normally do in a multi-well plate or a petri dish or anything like that. These microcarriers very often are made at 100 by 100 micrometers in size, which means that they can carry anywhere between 2 to 15 cells each, but we can change those dimensions if we want to. We also have a cell repellent wall. These walls essentially keep the cells contained within the peripheries and the boundaries of the microcarrier so that they can't grow off or walk off. The materials are also fully compatible with cryogenic storage. So once cells are attached on the microcarriers, you can put them in cryovials and freeze them down and thaw them again for later assaying. And then finally, there is a magnetic material that we've incorporated in these microcarriers, which allows you to immobilize the microcarriers, for example, at the bottom of a well plate, and also change their orientation so you can tilt them in 3D as well. What you can see on the right here is a single SemaCyte microcarrier with A549 lung cancer cells stained for actin and DAPI after PFA fixation. In this image, what we're showing is what the microcarriers look like when they are dispensed into a microplate and pulled to the bottom of the plate in the correct orientation. So you can see that they're all pointing in the right direction. And these are, again, A549 cells that have been stained with the AM calcine viability dye and with a nuclear mask. So these are our live cells that we imaged with live cell imaging. And as you can see, we can add less cells per well while still maintaining high local influency. And what this means is that we can ultra-miniaturize cell assays inside a micro well plate by dispensing less cells per well effectively. So why is this beneficial? First, we can now move cells during an assaying workflow. For example, after a drug incubation, you could collect treated cells which are attached to SemaCytes and move them to different wells to measure additional endpoints. So this allows you to produce more data per drugging event, all from the same treatment condition. The second advantage is that you can create large batches of cells on SemaCytes and then aliquot them into cryovials and create banks of assay-ready cells. This can increase your throughput because you don't have to trypsinize, culture cells, put them in a well plate, wait until they're attached or differentiated. You can just go to the freezer, thaw a vial of pre-prepared assay-ready adherent cells on SemaCytes, dispense them into a multi-well plate, and do your assay as you would do normally. It also can help you increase sort of the reproducibility of the data because you can make large stocks and batches of cells that were all prepared at the same time. And the third point is that we can, as I said earlier, reduce the number of cells per well, specifically by 5 to 50-fold, depending on how many microcarriers you add per well. And this is particularly relevant for assays that use primary cell materials or iPS-derived materials, for example. And later on, Dan will sort of show a couple of use cases of experiments that we've done in-house and that some of our partners have done with regards to these 3 points. So then moving on to the SemaPlex platform. This platform allows us to optically barcode the microcarriers. Specifically, we add optical barcodes to the walls of the microcarriers, as you can see in the graphic on the left, but also in the microscopy image on the right. So these barcodes are notches added to the walls. They function a little bit like a QR code, for example, and you can simply see them in the bright field of a microscope. The image on the right, what we're showing here is a pooling together of 4 types of cells, all loaded with a different tracker dye on differently barcoded SemaCytes. They were pulled together, added to a multi-well plate, and then imaged. So what's the benefit of this multiplexing? The main important point is that we can pool different cell models together into one well, so that we can do pooled cell screening in one well. Imagine, for example, that you're interested in screening 10 different cell models. Rather than doing 10 different cells in 10 different wells, you can pool them together with the SemaPlex platform and put them all into one well. So that means that you can save the amount of reagents, plasticware, and potentially time, depending on the size of the assay that you're running, by tenfold, which can be quite an intense and profound change. There's a couple of other things that you can do with the multiplexing. For example, you can add controls to each well for imaging, controls on differently barcoded microcarriers, for example, or you can also start to combine different tissue types in a co-culture setting, for example, to make deconvoluted organotypic models as well. What we're showing here is the composition of the microcarriers. So we're making the microcarriers with something which is called microfabrication techniques. These are the methods which are used to make computer chips, for example, as well in the semiconductor industry. This allows us a high level of control over their size, their shape, and their features as well. And as I said earlier, typically we make these microcarriers at 100 by 100 micrometers in size, but we can make them differently sized. We can make them bigger, smaller. At the moment, we make them as square materials, but we could also potentially make them as hexagons. So there is flexibility in what we can do through these microfabrication techniques. What you can see on the left is an array of immobilized SemaCyte microcarriers. So the way we make these and engineer these is we deposit them at the bottom of a glass slide, essentially. And this allows you to put on cells as per standard seeding protocols, something that Blaise will talk about a bit more in a minute. The cells stay attached to the bottom of the substrates until the cells are seeded on them. And then we have a smart release polymer that slowly dissolves in cell culture medium, dislodging the SemaCyte microcarriers, and then it allows you to trigger the release of these SemaCyte microcarriers with the cells into suspension to then collect them. We have a magnetic actuator. As I said earlier, this allows us to control the orientation of the microcarriers and also pull them down to the bottom of the well plate. And then the top sort of interface of these materials, essentially what the cells see is an inert gold layer, which we can attach with a variety of different cell attachment polymers. For example, fibronectin mimetic materials or straight up fibronectin, for example, as well, PDL, these kind of materials. On the right, you can see how we're deconvoluting the barcodes. So we have something which we call the SemaLyze software. And what this software is able to do is from an image from a microscope, it's able to identify individual microcarriers. It's able to read the barcode and then retile digitally all the images of the cells on a specific barcode together into one compiled image. And you can then take that image and feed it into any standard image analysis software. So this is how we're deconvoluting the barcodes here. And again, we'll show some examples of some of these experiments later down as well. The final thing I want to show here is how we are controlling the SemaCyte microcarriers with an external magnetic field. So you can see that the cells are all pointing in the same direction, and we can orientate them in 3D and deposit them at the bottom of a well plate. You can also see that these magnets don't clump together, so we can control their position without making them clump together, which is quite unique to the magnetic materials that we've included into this. So this was a brief summary into what SemaCyte microcarriers are and their features. And then now I will hand off to Blaise to talk us through the protocols.
Thanks, Jerome. Hi, good morning, everyone. So today I'll be introducing you to the SemaCyte products that we have developed. I'll be going through the general protocol for using them and highlighting some of the benchmarking experiments that we've carried out using A549 pulmonary adenocarcinoma cells. So SemaCytes are provided as arrays, which are immobilized at the bottom of standard tissue culture plasticware. There are two versions, the Seeding Dish 20 or SD20 and the SD75. And that has the standard dimensions of a multi-well plate, which actually makes it fully compatible with liquid handling robotic equipment. So the SD20 is a 20 centimeter squared seeding dish with about 50,000 arrayed microcarriers, allowing you to conduct 2 to 10 microplates worth of experiments. And the SD75 is a 75 centimeter squared dish with 200,000 microcarriers, which would provide about 8 to 40 microplates worth of experiments. And that's just depending on the desired density of SemaCytes required for your assay of choice. So as Joanne mentioned, standard SemaCytes have a 100 by 100 micron growth area and are coated with a fibronectin mimetic polymer, but there is some flexibility around the size and shape of the SemaCytes and the desired coating. We've been testing some other various coatings, and recently we've looked at PDL and fibronectin for human iPSC-derived cardiomyocytes. So we have a starter kit which contains all the essential equipment you need to begin running experiments with SemaCytes. The kit comes with 3 SD20s and 2 peripherals. The SemaPure 15 is used to purify the SemaCytes after cell attachment, and the SemaPlate is used to orientate and immobilize the SemaCytes, which have been dispensed into microplates. So the peripherals have been designed to sort of seamlessly integrate with your existing standard tissue cultureware and to enhance the experimental workflows. And they all come with the starter kit. So the protocol for using SemaCytes is quite simple and begins as many standard tissue culture protocols do. So the cells are seeded at the required density and in the required medium, and they attach to the SemaCytes, which remain immobilized due to that smart release polymer, which slowly releases over time. During seeding, the seeding dish can be kept on the SemaPlate to ensure that they will remain at the bottom of the dish during cell attachment. And then following seeding, you just move the dish into the incubator. So following cell attachment and the desired growth period, the cell-loaded SemaCytes are released into suspension just by gentle agitation and then transferred into a tube. So the debris and dead cells are removed using the SemaPure 15, which just quickly purifies and concentrates the SemaCytes, and that's due to their magnetic properties. And then from this point, the SemaCytes can be aliquoted into vials, they can be cryopreserved, and then they can be stored as assay-ready adherent cells. So the SemaCytes, they can be used straight after initial cell attachment or from frozen. So they can just be quickly thawed, purified, and then diluted to the desired density. They're handled in the exact same way as cells. So they're fully compatible with standard manual liquid handling tools and liquid handling robotic equipment. And custom magnetic SemaPlate holders can be designed to integrate with existing robotic liquid handling workflows. So the magnetic SemaPlate orientates the SemaCytes so that the cells are facing upwards and then pulls them down to the bottom of the well, after which the SemaPlate is no longer needed and the SemaCyte loaded plate is then ready for various plate reader and imaging-based endpoint assays. So for example, viability, proliferation, and mechanistic assays, to name a few. And the dual-sided nature of the SemaPlate actually means that the SemaCytes can be rotated so that the cells face downwards towards the microscopic objective. This allows for clear imaging of the cells on the SemaCytes within microplates. So to help you visualize the protocol a bit further, here's a 3D rendering and a bright field microscopy image of some arrayed SemaCyte microcarriers. So as you can see from the render, the cells are seeded. And then from the microscopy image, that shows A549 cells, which have settled and attached and allowed to grow for 24 hours before being labeled with nuclear mask and calcium to assess cell viability. So the solid SemaCytes are then released into suspension and purified using the SemaPure 15. And then they're dispensed into multiple plates using manual or automatic liquid handling devices, and the desired assay of choice is performed. So as you can see from the microscopy image, the cells remain contained within the walls of the SemaCytes due to their cell-repellent walls. So we conducted some benchmarking experiments to analyze cell loading reproducibility. We seeded A549 pulmonary carcinoma, adenocarcinoma cells onto SD20 dishes, and we used blank glass dishes as controls. So we seeded the cells at a density of 80,000 cells per centimeter squared and cultured them for 24 hours. After which we released them into suspension, purified them, and then dispensed them into 384 well plates at a density of 50 microcarriers per well. We then labeled the cells with calcium and nuclear mask and imaged them with the Image Express Pico automated imaging system. So as you can see, we see consistent cell numbers. Consistent cell numbers per SemaCyte when comparing SemaCytes arrayed in the seeding dish with SemaCytes which have been released in suspension. And we also see comparable numbers of cells across SD20 seeding replicates. So finally, for me, we looked at A549 viability after cryopreservation on SemaCytes as assay-ready cells. So in the top image, you can see single SemaCytes, which have been labeled with aim-calcine to determine viability pre-freeze and then post-thaw. So these were actually imaged 1 hour post-thaw. And as you can see, the cells, they've maintained an adherent and spread morphology on the SemaCytes and are labeled nicely with aim-calcine, which we quantified in the first graph. We also looked at the thaw recovery rates over longer periods of time and found recovery rates of 70% and higher for SemaCytes stored both at -80 and in liquid nitrogen. And we see comparable recovery rates when looking at vials frozen down from the same SD20 and also from different SD20s. We also used the PhenoView mitochondrial dye to assess mitochondrial health after thaw, and we observed healthy labeled mitochondria 1 hour post-thawing. So now I'll be handing over to Dan, who will go a bit more in depth into some of our SemaCyte use cases. Thank you.
Okay, so so Blaise and Jerome have given you an overview of the SemaCyte SemaCytes and also the standard workflows in terms of loading cells onto SemaCytes, handling the SemaCytes and freeze thawing. So I'm, for a number of use cases, really now going to talk about how the broad applicability to a range of cell-based assays SemaCytes are, and by using these assays, the advantages that you can get in terms of reduction of cell numbers, reduction of time, and I will do this by initially looking at 3 different plate reader-based assays, then talk about some imaging assays, how we can use more complex cells on the SemaCytes, and finally, the advantages afforded by the barcoding to cell panel screening. So our first use case is looking at faster viability assays. And if we look at the top left panel, this kind of highlights our standard workflow for using cells with SemaCytes, which is preparing batches of assay-ready adherent cells in cryovials. And for this assay, we would, for a cryovial, allow the cells to recover for an hour and then plate them out into a 96-well plate. For these toxicity assays, we've used a renal cell model in which we've plated out into a 96-well plate and treated with different doses of the cytotoxic compound ionomycin, and then read cell viability using CellTiter-Glo on a plate reader. And if you look at the dose response graph in the top left of the right-hand panel, what you can see is the cells on SemaCytes show a standard sigmoidal dose response to ionomycin. And importantly, the IC50 and the dose response curve are concurrent between using SemaCytes and then a standard well-controlled plate. And this really shows that by growing our cells on SemaCytes, you're not affecting their biology significantly or your assay readouts. We also ran this assay across 3 different centres with 3 different individuals, and consistently we got good concordance between the IC50 values for SemaCytes and well controls, and this really shows that the assays are easily transferable across sites and users. What's most important about this is not only are they usable in this assay, by using SemaCytes, you run the assay 2 times faster and used 6 times less cells. So I think this is a real advantage. The bottom images just highlight how these assays look and when you're on the SemaCytes and how the cells look. So this is an example of some cells treated with different doses of ionomycin. The green cells are live cells stained with aim-calcine, the red cells are dead cells, and you can see consistent with the curve, as you increase the amount of ionomycin, you reduce the number of viable cells and increase the number of dead cells. So the next use case kind of expands on this cell viability and cell proliferation approach. And in this case, we've used A549 cells, and the assay readout again is plate reader, but using CellTiter-Glo as a viability readout. And these are proliferation assays as we ran them over 72 hours. Again, we had assay-ready adherent A549 cells in a cryovial for the cells, allow them to recover, and then plate them out into a 96-well plate. And as we saw previously, with either trametinib, which is a tyrosine kinase inhibitor, or nuclein-3A, which is P53 stabilization, both of which has been shown to have cytotoxic or cytostatic effects on cancer cell lines. We have a dose response in terms of reduction in cell proliferation, and there's good concordance between the assay run on SemaCytes and well controls. And again, because we've not had to allow for the cell attachment time, because we can thaw the cells, they're already attached and ready to use in an hour, we save time within that workflow. The bottom image really just shows the flexibility that SemaCytes can give in terms of what you can do with them in assays, and particularly the ability to orientate the cells so the cells are facing up or facing down. In this image, we placed the SemaCytes with cells facing down, and in which case you allow the cells to spread off the SemaCytes onto the plate. And this could be used for longer term viability assays or pseudo colony assays, but also things like chemotaxis or migration assays. And this just really highlights the assay flexibility that you get with cells on SemaCytes. So the third use case, again, is a plate reader endpoint, but is looking at a mechanistic assay, and it's based on the NanoBRET system for looking at protein-protein interactions. And for the NanoBRET system, you need to transiently transfect the cells to run the assay, and this can lead to long preparation times and also limit the assay flexibility. With SemaCytes, what we've done is load the cells onto load A549 cells onto the SemaCytes, transfect them, allow time for the transfection to work, and then frozen the cell loaded SemaCytes. So we've produced a assay-ready adherent cell bank of NanoBRET transfected cells, and this assay we're using is looking at the interaction between P53 and its ubiquitin ligase MDM2. And with this assay, we again for the SemaCytes, allow them for an hour to recover, and then assay the BRET ratio at various time points after thawing. And if you look at the top right panel, what we can see is, firstly, again, the NanoBRET ratio is concordant between cells transfected on plates and cells transfected on SemaCytes. But importantly, also individual vials from the same batch show good consistency in terms of their NanoBRET ratio if they're assayed at 2 hours post-thaw or even 24 hours post-thaw. So this allows you flexibility within your time point for running your NanoBRET assay. As I mentioned, this particular NanoBRET pair looks at the interaction between P53 and MDM2, and this interaction is inhibited by the compound nuclein-3. And the bottom panel on the right just shows that the assay that we're using is acted, as we see the expected reduction in NanoBRET ratio when you treat the cells with nuclein-3. Importantly, by using SemaCytes in this assay, not only have we produced data faster, so 4 times faster, we've also allowed more flexibility for your protein-protein interaction campaigns. By reducing the prep time and also creating a large panel of assay-ready transfected cells, you really have ultimate flexibility on when you can run these assays, because quite often in campaigns, you will be limited to a specific day of the week that you can run that assay. And if you miss that time point, you have to wait another week to run the assay. Also, you may reduce assay variability by having this single batch of prepared cells for your whole campaign. So I'm going to move away now from plate reader based assays to talk about how SemaCytes provide advantage to image based assays. Before I go into details of a specific assay, I just want to look at the left hand panel, which shows how the images that you create from cells on SemaCytes can be processed and how they can be used with standard microscopes and image-based software. So on the left is just a microscopy image of the bright field and the fluorescent channels. We expect that most imaging systems would work equally well with the SemaCytes in-house. At the moment, we've used the EVOS and the Image Express Pico microscope. We've developed the SemaLyze software, which allows you to digitally identify SemaCytes. Once those SemaCytes are digitally identified, there's some basic image analysis within SemaLyze, so you can do nuclear counts and basic fluorescence intensity measures. But then also the SemaLyze software can tile the SemaCyte images. So if you look at the left panel on the top right image, this is a tile of the image on the left, produced by SemaLyze software. And once you have that tiled image, you can upload it into standard image analysis software such as Cell Profiler or ImageJ, and they will be handled in the same way a microwell plate image is. And this, the bottom right image on this left panel, just shows how Cell Profiler handles those images and can identify the nuclei in the cell bodies. We've also developed in-house Cell Profiler and ImageJ pipelines that again can just handle the raw images. So there's real flexibility in how you analyze your images, and it's basically similar to how you would analyze a normal microwell plate image. An example of the type of assay you can run is here. We've looked at immunocytochemistry assay, looking at p53 stabilization. We've tested a number of fixation methods and standard fixation method and not seen any negative impact on either the cells or the microcarriers by this fixation. In this case, we've used PFA fixation and used nuclein-free A stabilization. Nuclein-free A stabilization as p53 is the assay readout. And again, as you can see, there's good concordance between how the cells respond on SemaCytes or on microwell plates. But importantly, by using SemaCytes, you've produced the data 2 times faster. And there's also, by having the cells on SemaCytes and movable islands, flexibility within the workflow. We go into detail of that flexibility in one of our application notes on our website if you're interested in that. As well as immunocytochemistry assays, we've shown that standard cell painting workflows are applicable for cells on SemaCytes, and we don't see any obvious imaging artifacts or by using cells on SemaCytes. And by integrating SemaCytes into your cell plating workflows, you could potentially have a 20 to 2-fold faster data and use 6 times less cells. So that's sort of moving away from imaging endpoints to a lot of the data I've shown is working with cancer cells, but we also believe that more complex cells, such as iPS cells, would also be applicable for using on SemaCytes, particularly as these cells typically have a longer preparation time. So for the cardiomyocytes and the neurons we've worked with, you have like a 6 to 10 day preparation and recovery time required. And we've shown that we can load these BitBio neurons or axil cardiomyocytes onto SemaCyte microcarriers, and they can they express the expected markers of the cell type and the expected morphology. And by using SemaCytes with these more complex cells, which typically are non-proliferative, and because of that quite rare and expensive, you have a 5 to 10-fold time reduction in cell use, and also have faster assays. And we expect that these cell types would be able to be used in all the assays that I've described for cancer cells, and we're quite excited to look at all the different cell types that we can use SemaCytes with in the future. So the final use case is looking at how the barcoding that Jerome mentioned at the beginning of the talk can really lower the cost of cell panel screening. Cell panel screening is where you typically want to look at an assay endpoint across a large number of cell types. Typically, this can be anywhere between 5 to 20 cell types. In this experiment, we've done proof of concept for panel size, but we believe that you could easily look at 10 to 20 different cell types in a single well using the barcoding. And the workflow for this is you would individually load your cell types of interest onto 4 differently barcoded semaplex wafers, freeze them individually. So this is what we did here. And we used 3 different cancer cell lines, PC3, A549 and T98G, and the HepG2 liver toxicity model. Once we had our banks prepared, we thawed the vial, mix the SemaCytes together, so you have a mix of all the different cell types, and plated them into our 96 well plate. We then treated this plate with different doses of nuclein-3A, and then looked at p53 stabilization as the assay readout. And it's important to say that what we had is the 4 cell types in a single well, but we're able to produce single data deconvoluted for each of the different cell types. And the graph on the top right really shows the stabilization of p53 caused by nuclein-3A. As we would expect, the p53 wild type PC3 and A549 cells showed stabilization of p53 in a dose-dependent manner, whereas the p53 mutant T98G had high levels of p53 at the start of the experiment and didn't really show any significant induction of p53 levels with nuclein-3A. We see some induction of nuclein-3A in the HepG2 toxicity model, which is not surprising given that they're p53 wild type cell line. And we were able, by using the multiplexing capacity of SemaCytes, we've able to use 4 times less reagents and plate, use 60 times less cells, and run the assay 2 times faster than if you'd run a standard format. And obviously, as you scale up the number of cell types, those savings would be even greater. So I've now demonstrated all our different use cases and how you can save time and cells and reagents by using the SemaCytes. Jerome is now just going to talk about our early adopter program.
Thank you, Dan. Thank you, Blaise. So yeah, for the last couple of minutes, I will talk a bit about our early adopter program. So we've recently launched our products, our SemaCyte products, our starter kits, etc. as well. And we're giving access to these materials initially mainly through our early adopter program. And I hope that through all the different points that we've explained just now, we were able to highlight sort of the value we can provide with the SemaCyte platform in terms of how easy it is to put cells on the microcarriers and how easy it is to reap the benefits of these materials for any standard sort of assays and workflows, all the way from plate reader based to imaging workflows, and really how we could potentially with these materials reduce the time and cost of assaying campaigns, and also make workflows much more flexible, which is always quite interesting. The program gives you access to SemaCyte materials. So essentially, we give a free starter kit available and discounted SemaCyte seeding dishes at a 60% discount. We're doing that for the period of January to March. We ask people to apply for this program by December 1st. And then if you're selected, we will come over to give training in terms of how to use the materials, and we'll also give ongoing support, which will give you the opportunity to validate an assay of choice and integrate that with your workflow specifically as well. So if you're interested, either drop us a mail, happy to tackle any questions that you might have with regards to this, or just go to our website and just sign up for the program, and we'll send you more details about that as well. I have to say that beyond the early adopter program, we are also setting up collaborations and joint development projects. This is specifically for some more complex work, for example, using the multiplexing. One example is we're setting up an arrayed CRISPR screening workflow using the multiplexing with a collaborator as well. So if there are more complex things you want to use the SemaCytes for, feel free to reach out, and we could potentially also discuss a collaboration or anything like that as well. That was everything from us. So if there are still any questions, feel free to pop them in the Q&A section. And for the next 10 to 15 minutes, we're happy to take any of those questions. I'm already happy to tackle a few straight away. So one question is what the maximum amount of barcoding is with regards to the multiplexing and how many different types of cells you can theoretically combine. Our barcoding protocols allow us to generate millions of unique barcodes. However, it will be impossible to combine millions of different cell types into a single well. So we believe that the maximum amount of multiplexing you can do in a single well would be about 10 to allow you to have enough cells per well to have good statistical significance. So I think 10 cells per well is the maximum that we can do for barcoding. However, we can create millions of unique barcodes if that would be of interest. And some people have been interested in using SemaCytes that all have a unique barcode to, for example, trace the behavior of specific cells on SemaCytes, but that's a whole other project in and of itself. Another question that we got is how many times you can move the SemaCytes, for example, and the answer is that, yeah, you can move them as many times as you like. So once they are in a multi-well plate, you can collect them, for example, after a drugging experiment, and you can redeposit them or redispense them into other wells, for example, to do more endpoint experiments. But even after that, you can still recollect the microcarriers and do something else with them. This may be tangential to another question with regards to RNA sequencing. So you can do RNA sequencing, for example, on these materials. You essentially just trypsinize the cells, collect the RNA, do your experiment. So you could, for example, do a drug incubation experiment, collect a sample of cells and SemaCytes that you use for an imaging endpoint, but in another sample you can use, for example, for a RNA-seq endpoint or something like that as well. Let's see, some more questions. So we got a question with regards to different types of coatings and what we aim to provide with regards to that specifically. So what we're doing right now is our basic materials have an in-house engineered extracellular matrix mimetic peptide. So there's a fibronectin mimetic polymer that we've put on the SemaCyte Microcarriers, which has been working quite well for most standard cell lines. However, for some of the more complex cells, such as the bioglutamatergic neurons and the cardiomyocytes from Axol, we've been using other types of materials, such as Geltrax and fibronectin. So it is possible to put these other molecules directly onto our fibronectin mimetic materials. So that you have a layer of human fibronectin, for example, on top of that, or Geltrax, so that's possible. And there is flexibility as well to provide bespoke coatings straight away. But end users are able to add another layer of coating on there if that's something they would request, for example. So then there was more on the imaging and on the software itself. So there was a question on whether or not the microcarriers can be analyzed through, let's say, Harmony software directly, or whether or not they need to be extracted and added to the SemaCyte software first. So if there's no barcoding involved, there's no need to extract these images and retile the SemaCytes into an image that's then analyzed downstream. Any sort of image analysis software, whether or not it's the Harmony software from the Opera Phoenix or so, can do that automatically. When the barcoding is used in the experiment, at the moment we need to extract the images and retile them into their specific barcodes for analysis. Otherwise, we cannot do the deconvolution. But we're working with some of the solution providers and trying to make sure that we can integrate some of these software bits as plugins in, for example, specifically the Harmony software. So that's something that we're working on at the moment. Couple more questions. There was a question on the neurons and the size of the microcarriers with regards to the neurite outgrowth. So these microcarriers are 100 by 100 micrometers in size. So at the moment we get around 5 neurons per microcarrier. So it doesn't allow for a lot of room for the neurites to grow if you want to do neurite outgrowth assays. we are designing different microcarriers which are larger or have long shapes in which the neurites could do some more growth, for example, as well. But likewise, this is something that Dan highlighted. You can also deposit the microcarriers with the cells so that they would interact with the well plate. And then the neurites could grow off onto the well plate, for example, even interacting with neurites from other SemaCyte microcarriers as well. So there's a lot of flexibility in the system in terms of how you can design your assay and what kind of experiments you can run. Take maybe 2 more questions or so. One was, quite related to what I said earlier about the neurites growing out of the SemaCyte microcarriers onto the surface of a well plate, for example. There was a question on whether or not you can do migration assays with the microcarriers as well. That is possible. Something we haven't done ourselves internally, but anything like a migration assay or a scratch test type assay, we believe should be possible, again, by having the SemaCyte microcarriers face down on the well plate so that the cells can grow off the microcarriers. So you can imagine a system in which you put different types of coatings or dead cells or anything like that onto your multi-well plate and see if there's any difference in sort of the migration of the cells out of the microcarriers, for example, as well. We also got a question on electroporation because Dan highlighted that we can use lipofectamine to transiently transfect cells on SemaCytes. So we have validated that that's possible. We haven't done any work ourselves with regards to electroporation. However, we're setting up a couple of projects and theoretically there should not be a limitation with regards to this. So you can imagine a system in which you have cells attached to SemaCyte microcarriers, fully adherent. You have these in suspension and then you can use electroporation to get whatever your constructs are inside the cells. Some people have highlighted to us that that would be of value because at the moment, if you want to do electroporation, you have to trypsinize your cells. The cells have to be in suspension. So with our system currently, we could actually try to electroporate fully adherent cells. And again, this is useful for iPS-derived models, for example, that take a while to establish and that you definitely don't want to trypsinize. So this is certainly something which is possible. The last question I'll highlight is on flow cytometry. So somebody asked us about flow cytometry. This is a question that we do receive quite regularly as well. We haven't tried to put these microcarriers in a flow cytometry device, but the idea there would be that you can get your cytometry data from fully adherent cells on microcarriers. It's something that we will be looking into in the future. It might be that we have to make the microcarriers a bit smaller because you're limited by the size of the capillaries in a cytometer. Very often they're, I think about half a millimeter in size. I can't remember the exact dimensions, but we have to probably play a little bit with the dimensions of the microcarriers to make sure that they fit in there. And then there's the other question of how many cells per SemaCyte you want, because if you have some SemaCyte microcarriers with 1 cell and some with 2 cells, for example, that's going to make your cytometry data analysis a bit more complicated. So I think in an ideal world, you would want a system where you only have 1 cell per SemaCyte. And again, this is something that we could develop in the future, but something we haven't done so far. I think I don't see any more questions. So if there is still something anybody would like to ask, feel free to pop it into the list of questions. I see there was another question that I overlooked. Yeah, okay, we tackled that question already with another question. So I think that is everything there. So yeah, I think happy to conclude everything there. Again, thank you everybody for attending our webinar today. It was nice to share with you all what we've been doing over the last year or so in terms of the development of our SemaCyte microcarrier platform, to share with you how you can use these materials, what kind of assays you can use them for. And we are quite excited about getting these materials now into into the labs to accelerate some assay workflows. And again, if there are any thoughts you have, any questions, or if there are any assays that you think we should be validating in the future, feel free to send us a message, and yeah, we'll have a chat. Bye for now. Have a lovely day, and hopefully see you soon. Goodbye.